Welcome everybody to Carney Conversations. It's really a thrill to be here with you again. This event today is entitled The Next Generation of Scientists on the Future of Brain Science. And Chris, who I'll introduce in a second, and I are just really thrilled to kind of host this particular event. And I think we know intuitively that if you want to know what the future is of brain science or any field, you invite the graduate students and ask them what they think, because they're like way more interested and creative and innovative and seeing connections where it might take Chris and I a long time to make those connections. So we're really tremendously excited to have you all here. Chris Moore is the Associate Director of the Carney Institute, and so he and I have been doing these events for some time now, and they've been really incredibly fun. So I'm going to do a quick introduction to our guests, and then you'll see that they're just going to take the show away and engage in a really interesting discussion about how they got to where they are and what they see as the future of brain science. So quickly then, Kaitlyn Hajdarovic is a PhD candidate. Hey, Kaitlyn, say hi.
Hi.
So Kaitlyn's in the Neuroscience graduate program, and she's pursuing research in Professor Ashley Webb's lab. And as you will hear, Kaitlyn's developing novel cell reprogramming platforms to study neuronal aging, incredibly topical right now. And Kaitlyn received actually a Carney Graduate Award for her research, and you are highlighted, I think, on our website. It's a great piece about you and your research in the web lab. So we'll talk to you in a second, Kaitlyn. Jae-Young Son, hey, Jay, there.
Hey, everyone.
Jay's a PhD candidate in the Department of Cognitive Linguistics and Psychological Sciences, and Jay is working in collaboration with Professor Ariel Feldman-Hall. Jay studies how we navigate the great blooming, buzzing confusion of the social world. Jay has a fantastic kind of blog. I spent a little time like cruising that, super fun and exciting, but of course his research is again also very relevant right now. So we'll hear from you shortly, Jay. And our other guest is Marc Powell. Hi, Marc.
Hi.
Marc is a graduate. He's on the other side of the PhD dissertation. He graduated this year from the Brown PhD program in biomedical engineering. And he worked alongside David Borton. And what Marc did was to build these implantable devices to record neural activity, and also to stimulate brain or spinal cord to modulate neural activity. So you can see the breadth of the research that we're covering right now with our guests. Marc currently is now a postdoc associate at the University of Pittsburgh in the Department of Neurological Surgery. So that's everyone. So the way that we're going to do this is Chris and I are going to ask a few questions, and then at any point, please, audience out there, feel free to post any questions that you might have in the Q&A, and then Chris and I will kind of look through those and send some questions to, or ask the questions of our guests here. And the questions are only visible to you; they're not visible to the general audience. But we're going to pick and choose. But we have a bunch of things to get started, and I'm just going to kick off, and then Chris and I will be going back and forth. But just wanted to maybe Kaitlyn, we'll start with you. I think we all enter research. We all have our own paths and have our own story about how we got to where we are right now. But I just think it would be really fabulous to hear from you all. What was your path? How did you get to where you are now? Kaitlyn focused on kind of the biology of aging. What was your journey?
Yeah, so my journey was kind of a winding path. I majored in neuroscience as an undergrad, but I thought I wanted to be a clinical psychologist and, spend my day talking to people. I ended up working in a lab when I was abroad in Australia at the University of Queensland. And I was on a project trying to generate a screen for cone snail venoms to see what kind of mammalian receptors they might bind to for potential drug discovery. And that was kind of, it was a moment like a lightning bolt hit me, where everything I had learned up until that point that I kind of thought was like useless and maybe not interesting, like all the molecular cell biology stuff that I was like, I'm never going to need this. It all suddenly made sense, and it all suddenly seemed like the coolest thing that I could possibly be working on. So that's kind of when I made the decision that I knew I wanted to go into kind of wet lab molecular biology type research, but still focused on the brain. And then I got into aging when I rotated in Dr. Ashley Webb's lab, and I was, I think everyone who studies aging kind of has a personal insight into this. My grandfather had Alzheimer's disease, and I was very intrigued in that disease, but maybe from what I had read, discouraged about the way we have been studying it and the potential for treating it. And learning about the kind of genomic approaches in the lab and the way we are able to generate, like human cell types in a dish and study those, I was kind of like, this is where I want to be. And I think this is the path forward for, curing these diseases. So that's how I ended up there.
Thanks, Kaitlyn. I love that you just really brought us into your world in that moment of, oh, this is what research is about. And, just like seeing it and living it. It's really fantastic. Marc, how about you?
Thanks. Yeah, so my path has similarly been kind of circuitous, but I'd say I've always been interested in engineering. I've kind of always known that that's what I wanted to do. My path towards the brain science side of things definitely took a different turn. So actually, if you go back far enough, I wanted to be an aeronautical engineer. I've always been sort of interested in flight and this idea of being able to go wherever you want and things like that. But actually in high school, I had a biology teacher, Stephen Fowler, who unfortunately passed away when I was in undergrad, but he really made just this really lasting impact. He had this sort of undimmable excitement about biology and about science, and it was really infectious. And biomedical engineering for me really gave me this opportunity to merge those two interests. And then while I was in college, maybe perhaps slightly less gloriously, I was fascinated by this idea of being able to control things with your brain, science fiction, Star Wars kind of thing. And that led me to the path of brain-machine interfaces, which really is, in my view, this science fiction realm come to life in a lot of ways. And so that was the path that I chose to follow. And I got really, really fortunate that when I applied to join at Brown, there was positions that were open with some really, really talented engineers and scientists. And that's kind of how I got to here as well. And now in my postdoc, I'm actually fortunate to take what I did in my PhD, which was really engineering focused, and transition that and actually work with people on a day-to-day basis. And that's been really, really rewarding.
Thank you, Marc. Jae, do you want to continue the round? Thanks.
Yeah. So I study social networks and how people create mental maps of social networks and how we navigate through social interactions using the maps that we create in our minds. And if you look on paper, at my CV, which details all the research experiences I've had, it looks like I've taken a pretty straight shot to getting here. But nothing could be further from the truth, right? Like the other panelists, I've taken a really windy path to get to where I'm at. So for me, it really actually started when I was a kid. I grew up in Tennessee. And while most of the social interactions I had there were totally fine, of course, there were some people there who took note of the fact that I am a racial minority, who had racial prejudices and racist beliefs. And so for me, it was a problem of actually creating these mental maps of who to ally with or who to avoid in my social interactions, right? And that actually got me started in this whole line of questions really early on. Well, in high school, I actually had thought about dropping out and becoming a professional musician. And it was really the urging of some high school teachers of mine to push me back towards scholarly pursuits. I told my parents, whatever, I'll go to college and I'll do the thing. But, just so you know, this is just a backup plan, right? I have an idea for what my day job's going to be, and it's not going to be, So I went to college, same sort of thing. Even though I was working in the research lab, and was seemingly doing okay in all my classes, in the back of my mind, I was always thinking to myself, is science right for me? Do I belong here? I found myself struggling in quantitative or methods classes. And I thought, well, if I can't do these things, then, do I really belong in science? I kind of stumbled forward into a lab manager position here at Brown, working with Dr. Oriel Feldenthal. I got to continue working on this line of questioning, start to really develop my research interests. And this is around the time when I realized that, like, for me, the thing that makes science really fun is that for me, it's totally personal for me, right? These are questions that impact me and that allow me to make sense of the experiences I've had in my life, but in this really systematic sort of methodological way, right? Where I'm trying to uncover basic truths about the way that we approach social interactions and how the brain subserves our ability to navigate the social world. And so for me, science is not a spectator sport, right? And so that's really what keeps me going and being who I am now.
Yeah, I'm gonna flip over to Chris in a second, but it just really strikes me with each of your descriptions about how you got to where you are right now, which is just the beginning of what you will be doing. It's just really personal, right? Like this searching for this point of satisfaction, bringing your experiences, and then just having, I think for Kaitlyn and Marc at least, it's like, oh, I can control this. Like I've like something I want to do, and I can actually do it. And for Jay, there's this degree of uncertainty about what outcome there might be, depending on kind of social context. But I think, Chris, do you want to? I'm going to flip over to you because I know that you have a lot of interest in how to do science and how we do science now, and what the future might look like.
Sure. Yeah, when continuing what Diane was saying, you all captured really nicely something that's often the actual way to endure all the crap you have to deal with in science is if you just have that thing that you're really, truly curious about, interested in, from toxins all the way to brain control to social network, like whatever that is, when you have that, then you always know you're working on, it has this intrinsic reward. Even when you learn, again, negative result, it's not, oh, great, now I know it's not that, like it has that reentrant value. So give us a sense of your work. And we got a little bit from Jay, but maybe Kaitlyn, like maybe give us a little bit more of a dive into what you're working on. And something else maybe to throw in there that I think is really neat to talk about, as Diane said, I'm sort of addicted to thinking about questions like this. Is there in your kind of science, is there a sweet spot for the kinds of groups of people you need to do it? Like some science, there's this myth of the totally isolated scientist, and there's also the myth that it is only by the full community. There's something about the individual, and there's something about groups too. And maybe give us a sense from the kind of work you do. I don't know, how many people is the sweet spot for a team? And
Yeah. So I am interested in a brain region called the hypothalamus. And obviously I'm very biased. I think it is the coolest brain region. It controls things that are essential to survival. So sleeping, eating, water intake, blood pressure, even a lot of social behaviors like aggression and sexual response, hormone release. It's pretty much everything that starts to go haywire with age is, the master regulator for that is in the hypothalamus. So my interest is understanding, like what kind of individual cells are involved in these processes and how do they change with age. And I'm also interested in coming up with better ways to study this brain region. So people have been kind of fascinated with the hypothalamus and aging for a while. Like, this isn't new, but we've only very recently started to develop the tools that are really necessary to actually dive into this brain region. So the hypothalamus has a ton of really specific types of neurons that all secrete different factors and control different processes. And understanding, like pulling out those individual cell types and understanding what they're doing, kind of up until now has been super difficult. So I use a platform called single cell RNA sequencing, and you can look at gene expression in individual cells with this platform. And I'm looking at how these cells change between young and aged animals. And also with that, Diane kind of referred to my reprogramming project. I am trying to generate basically the aged hypothalamus in a dish. So there's a unique hypothalamic cell type called the POMC neuron. And this neuron is part of a system that basically decides when you eat and when you're full. And it lets you know, like, you have enough energy, you don't need to eat anymore. And obviously, body composition changes a ton with aging. So I want to understand what these cells are doing. And there's not that many of them in a brain. It's hard to get neurons out of a brain to study them ex vivo. So I'm using a method called direct conversion, where I can take a skin cell, a fibroblast, and overexpress certain genes and generate a brain cell that will have the aging signature. So it has DNA damage, it has messed up mitochondria, it looks like an old cell, and it goes from being an old skin cell into an old neuron. And I'm trying to create, basically make old POMC neurons in the dish. And for me, science is a team sport. So I, some of the stuff I do, like day to day, if I'm in the TC hood, that's by myself. If I'm analyzing my data, that's by myself. But everything I do, I tend to be very collaborative. So getting my single cell project off the ground, I was collaborating with a couple different labs to learn how to do the process, learn how to do the data analysis. And then, my single cell project is a small paper right now. And the amount of people we actually need to get this paper out the door is, it's never going to be one person. It's not even going to be 3 people. It's a lot. And I actually really like that, because I can never know everything and I can never be an expert on everything. But when I'm pulling together all these experts, that's what makes the paper and the research fantastic. So that's my take.
Awesome. Yeah, I think you hit on something that's great to highlight too, that especially if you are investigating truly new areas, some degree of making a method, tweaking a method, adjusting a method, stealing a method from a way it's applied somewhere else, which is a great way to get credit for making one. It's always part of it. Thank you. Marc.
Yeah, so a lot of my PhD work, I actually spent more on the tool development side than Kaitlyn described. So I've spent a lot of time thinking about the engineering behind neuroscience, and how we can actually access parts of the nervous system. So if you go back far enough, early on, we were recording neural activity with single electrodes, one at a time, where we'd probe these into the brain and we'd listen to the cells. When I say listen, I mean we're listening for these action potentials that the cells are participating in, which is one way that neurons communicate with each other. Over time, we've built up our tools to be able to record from more and more of the brain. And for the past couple decades or so, those exist in the form of 100 or so different electrodes that are implanted in the brain or other parts of the nervous system. And that is better. It allows us to record multiple neurons simultaneously. But there's this question of, well, okay, these devices are 4 millimeters by 4 millimeters. So if we're recording from such a small area of the human brain, which is such a big volume, what are we missing? It's incredible that we can learn so much from such a small area of the brain, for sure. But there's this question of, in the engineering side of, well, how can we get access to the whole nervous system, or even maybe less ambitiously, just several thousand neurons at once? And what can we learn from that? So my PhD really was spent trying to ask that question about what are these engineering design problems and challenges that we have in order to access lots of the brain. And my work at Brown really focused on being able to access simultaneously with these 100 electrode arrays, but from lots of different parts of the nervous system at once. So we can look at how do these areas communicate with each other. And with the goal of being able to do that with, say, 10 or so arrays, and therefore being recording from 1,000 or so. And now there's companies, and there's a huge effort from the government and from lots of industry to record from many thousands or millions of neurons. And I think that's really the way things are going, but it was exciting to be part of it and get the experience I did from my graduate work. In terms of the effort that goes into a project like that, I think to assign a number to it, I'd be putting sort of an arbitrary value, and I'd probably be not doing justice to the many, many supporting roles that go into projects like this, especially in clinical research. We have, the hospital administration, the university administration, physicians and everything like that. But from a purely technical standpoint, sort of the technical team that are involved in projects like this, especially ones that are involving human subjects, you have to have medical experts, you have to have engineering experts, you have to have science experts. And then when you get into actually decoding from thousands and thousands of neurons, you have to have computer science experts and algorithm experts. And so it really is this hugely interdisciplinary team that makes this all go round. And I think when I came into my graduate degree, I thought, as probably many other neuroengineers do, that I'd be able to kind of tackle a lot of these challenges on my own. And I think it is the nature of grad school a little bit, that you kind of get stuck in this situation where you feel like you have to do all of these different aspects of a project. But what's really inspiring about neuroengineering is the ability to work with so many other talented individuals. And certainly that's not to take away from the fact that a lot can be done by an individual. Even without a whole bunch of support, you can learn a lot on your own, even if you're not a graduate student. I think neuroscience is one of those fields that can be really accessible, surprisingly so, considering how complex the brain is. But there are tools out there for it. So I think that's my maybe slightly indirect answer, but hopefully, it got to this idea that science in neuroengineering is a hugely collaborative effort, but there's really a lot of talented people who individually contribute quite a bit to the research.
Yeah, I really like that you made a point of emphasizing too the real incredible iceberg. And when we publish scientific papers, there are a number of people listed on the paper, right? But that really is exactly like an iceberg. There's a 90% of people without which there's no university, there's no process, there's no support, and it wouldn't have happened. Yeah. That would never be listed on a paper under the conventions of how we do publications for the most part these days.
Yeah, and I'd actually like to add to that point exactly that, especially in human research, I have found that some of the most talented, most sacrificial people are the people who actually are subjects in these trials. And they actually don't often get published as authors on a paper, which is probably a bit of a shame. I mean, these people are donating their time and their lives in a lot of ways to this research just so graciously. And honestly, it's really, really inspiring to work with those people because, their dedication to science is one of uncertainty, right? They don't know what the results will be for themselves. They're really doing it for the benefit of others, 10 years down the line or something. And that's really inspiring to see.
Yeah, there's this historic allergy to that, right? Partly because of early psychophysics, to be nerdy about it. And that historic allergy has not aged well. I mean, especially with the reality that every paper that's written is written from someone's perspective inherently, as if that wasn't true. So why not include all the people that, especially the subjects? I wonder if there's some fraction, like number of people per number of neurons, and there's a ratio. We can come up with it. We call it the Powell power law or something. So Jay, tell us about your work and maybe about groups, sizes that can do it.
Sure. So like I said before, mental maps of social networks is what I'm all about. And just give you a flavor of some of the questions that we're asking lately. So the problem with a lot of social networks is that they are way too big to be learned. And so it's more or less impossible to actually create a mental map of social networks, which is terrible for me because that's my whole research program, right? No, but like, with every network member who's added, the number of possible relationships between people increases combinatorially. What that basically means is there's a whole explosion of possible relationships, right, that could exist in a network. And yet, most of the time, we don't have direct exposure, or we're not able to observe those relationships taking place. And so this is a real problem, because if you're trying to create these mental maps, you're going to end up with a whole lot of biases depending on what you can observe from direct experience. So some of the questions that we're asking now is, well, how do people fill in these gaps, right? And so there are just so many different levels at which you can start filling in gaps. There's some exciting work that tries to preserve the identity of individuals in a network, and to just say, can I take something that I know about the topology of networks, that people tend to form these groups, or, a friend of a friend is also likely to be a friend of mine, or you maybe have heard it the other way around, which is the enemy of my enemy is my friend, right? These are certain principles that we can use to try and fill in these gaps. I'm also interested in how we use what I call features of people to fill in the gaps, right? And so, for example, if I go out into the world and I observe frequently that the chemists here collaborate a whole lot with the biologists, then that gives me some basis for inferring a friendship between some people I might not know. So there's a professor of biology, a professor of chemistry. I have no idea whether or not they collaborate with each other or they're friends, but if I know that people in my university, let's say, tend to have that relation in abstract feature space, then I can infer these relationships without ever having to meet these people. I abstract individuals into these features, and then I learn relationships between features, right? And so these are different levels at which we're thinking about this problem of filling in the gaps in our knowledge of social networks, of how we make these maps. And so, when you talk about the sweet spot for teams of people doing science, isn't that sort of like the holy grail of managerial science, is finding the right size of team so that you can, really extract out the most creative, most best performing thing from, the fewest number possible. And so this actually, this is a bit of a tangent, so you'll have to forgive me, but this reminds me a little bit of the Beatles, right? There's this famous quip from John Lennon when he was asked, do you think Ringo Starr is the best drummer in the world? And he goes, no, I don't think Ringo Starr is even the best drummer in the Beatles. And then it's interesting to contrast that against with what some of the Abbey Road sound engineers would say, which is, if you can get a few of the Beatles into the same room at the same time, that's really great. They're all enormously talented musicians. But on the rare occasion that you can get all four of them in the same place at the same time, something just extraordinary happens. There's a synergy where the productivity of a collective of people, the right people, produces something so much greater than what any combination of them could produce. And I think really that's what happens in science. The other panelists also talked about science being a collaborative endeavor of these massive research teams needed to make any sort of progress, and so much of it goes unacknowledged. And so it's funny for me to think about like all the Ringo Starrs in science. We need them, right? And to think about, well, do the Ringo Starrs in science get enough credit for the supporting role that they play? That seems, so whatever, it's a supporting role, but it's essential, right? We can't do science without it.
Yeah, and the role of the engineers as tying together groups of people by not having to be the musicians themselves, but you couldn't do it without them. And literally billions and billions of people never would have heard the Beatles without. So this idea of their researchers and support is also an antiquated notion. It's really just a big network. It sounds like your research has the key to how we should, come down to Carney and tell us some principles of the right way to structure a social network, because it's far beyond managerial science, I think, in that sense. Really
well,
cool.
One of the things that makes Carney such a unique and fun place, right, is that it is actually a space for people to come in and do exactly that, right? I mean, why do we have universities, really? We could have independent contractors of all the researchers out in the world who are the best at something. They all have expertise in whatever they might have. And so, you know, you need somebody to run some sort of analysis that you don't know how to do. Why shouldn't you just go to, you know, some website, find somebody who can do it, and then contract them out for the job? Why do we stick to this university structure where we try to build communities of people, right? So that you can, maybe you don't know somebody who can do that analysis, but you know somebody who knows somebody who can do the analysis. And I think that's really the value of having people together in a community, and the sort of challenge, I guess, that you and Diane face in trying to build community in the Carney community. But that's really how the free exchange of ideas and the cross-seeding of perspectives happens. And that's where innovation really, I think, emerges.
And if I could add to that, I think COVID has really shown how essential these communities are. So during COVID, we were kind of doing the almost very impersonal, like, oh, I need something, I'm just going to email them. We didn't have those, oh, I'm talking to my, you know, people on my floor at lunch and talking about a problem, and you're missing that kind of like magic spark of, we're just going to get together and chat and come up with these, you know, amazing, beautiful things or find solutions to these problems. And doing science in this kind of very narrow, just by yourself way has been a lot more difficult than I think I would have anticipated.
Mm-hmm. I mean, it is the experiment none of us wanted.
Yeah.
Just sort of like, do we really need to be interacting in person, and do we really need those spontaneous moments, unplanned? And I'd vote yes. I mean, Kaitlyn, you just said absolutely categorically, that's how we function, that's our oxygen. But yeah, none of us would have wanted it that way. But I agree. Jay, I kind of wanted to come back a little bit and then engage everybody in this. You said something, I was kind of picturing and asking myself the question, which of course for me, I know what the answer is, yes, but like if the Beatles appeared now in this moment in time, like would they be, you know, such an incredible band? Like, so the point is that you need the right group, but you need the right group at the right time and in the right moment. And so I wonder how you all think about, perhaps we'll start with Jay, because you so it's like impinging on your research, but more broadly, you know, as we think about, I'm going to work on this, I think it would be really helpful for the audience here is to, like, how do you like make that choice? Like, how much of what's going on in the outside world, how much are you generating internally? And then how do you think about those two things interacting? And I guess the point is that many people work on something at completely the wrong time, at least for them, for them to be known for something. But then, of course, later we're all delving into the literature and looking at what's been done, and then that convergence of things you can't control, but drawing from that building of knowledge. Anyway, I think there's a question there, which is, but I don't know if Jay, if you've thought about this in terms of your own research, how do you build in that time component, that moment in time? And then, you know, for Kaitlyn and Marc, like, what are the external influences that have really worked on you to make you pick your area? So, Jay.
Yeah, I mean, this is such an interesting question. I think about, well, this is something I didn't realize before I started doing science, which is that so much of what we do in science is just have a conversation. We're holding dialogue with our contemporaries, with some of the classic historical thinkers who put forth these big ideas. And so in that same way, art is, you can think of it as a dialogue, right? There are people who go out and try something new. There are people who react to it, the pendulum swings this way and that. And then that produces, over time, what we see in retrospect as movements, right? Or periods or eras of art that are distinct and creative and have their own flavor that's different from other eras. And I think the same can be said of science. It's very emergent, so you're absolutely right. you can take a group of great people and put them in the wrong environment, and maybe that stifles some progress. But I think the thing that's interesting about thinking about social networks is that it forces you to think about ecologies as well. So what I mean by that is, if you have a community of people in an ecosystem, you can ask those people in the network to adapt to their surroundings or to their environment or to their context. This is adaptation, one of the fundamental mechanisms of evolution. But there are other aspects of ecosystems thinking that I think we often overlook, which is that members of a community or the communities themselves will actually go out and reshape their environment to match whatever their needs are or whatever their goals are. And I think it's true that if the right group of people in the wrong place or time, you might not get as good of a result, but those people are in dialogue with each other, are creating the social conditions necessary for them to pursue discovery and to pursue innovation. And so my sense for things is that if you get the right people together who have a shared vision or a shared goal, that they are going to reshape whatever their environment looks like, whether it be the members of their community, so shaping the network itself, shaping the resources available to them by looking for opportunities, and they're going to largely be able to do the same things, maybe just in a different timeline, with the obvious caveat, of course, that if you're not funded, you can't do science, but such considerations aside.
Any other thoughts?
I'll actually, yeah. So I found myself entranced by Jay's description there, and actually, there's a couple kind of things I'd like to add on. So I guess there's this question of who is the right person for a job at any particular point in time, and I think there's two factors that play into someone being really excelling at a particular role. The first is the skill. They have to have the abilities to do what's required of that role. But the second is the motivation and the sort of excitement and the drive to do it properly. And we talk about ourselves as neuroscientists or neuroengineers or brain scientists or whatever. And the thing that binds us all in that particular description is the brain or the nervous system. But there's, I think, another way to look at that in neuroscience specifically, we're trying to answer such challenging questions that the right person for a neuroscience question might not have ever thought about a question to do with neuroscience in their lives. And I think a really good example of that is one that's perhaps a bit controversial in the neuroengineering realm right now, but Neuralink, which is Elon Musk's new company, they took this approach where they reached out to the engineering community as a whole and said, we want to build devices to go into the brain, but we don't care if you've ever thought about neuroscience before. What we want are people who are experts in engineering these specific types of things. So they looked at cellular engineers, people who build cell phones, who are really talented at this radio frequency data communication and can make data go places really quickly, or ASIC designers, so chip designers who are really talented at putting transistors on a chip in just the right way to make them super efficient at what they do and perform really well, analog electrician, all of these people who and they kind of put them all in the right place at the right time. And I think that was the right mentality to build what really is impressive hardware. And I think the controversial piece is now they're struggling to figure out exactly what to do with that scientifically. But maybe that just means that now it's time to recruit the right people for that task. And so I think that's one really interesting aspect of science as a whole, and neuroengineering specifically, is that it really is people that it just requires the people with the right skill and then the desire to answer the questions in neuroscience. And I think that's how you build that team. That's easier said than done, but.
I think what you said about the Neuralink issue right now being like a tool in search of the problem, that's something I find very interesting, because doing single cell RNA sequencing, it's a very trendy kind of analysis. And I have been very curious about how people kind of shape their research based on the tools at hand. So, if all you have is a hammer, everything looks at it as like a nail. If you have single cell RNA sequencing, you're gonna look at a million cells. And I think, when you're thinking about who's doing the research and how they're doing it, the best researcher is the one who's going to ask the right questions. And I think that can sometimes be the hardest part, because that requires understanding of the literature. It requires understanding of who you have to work with, and it requires understanding of tools that might be changing as you're doing the research. I've recently had this really kind of interesting experience where I did a single cell RNA experiment a couple years ago. I recently redid it to, improve my numbers for a paper, and the technology has, I feel like I put my head down to do this paper and I looked up and the field is all the way miles away and I'm running to catch up. And so I think, being able to adapt and stay on top of things is also just a huge part of how science gets done.
Kaitlyn, do you want to just add a little bit of explanation about why would you want to do a single cell RNA seq? What does that give you? Why not just the brain?
Yeah, why not just the brain? So this is something that I find, this technology, applying it to the hypothalamus specifically, has been really fascinating. So single cell RNA sequencing, as you can imagine, is able to look at gene expression at mRNA at the level of the individual cell. And in a tissue like the brain, where all of the cells are very unique, they're all doing their own thing, secreting their own factors, responding to specific stimuli, being able to look at just one cell or one group of cell or one type of cell in a specific context is extremely powerful. You're looking at these signals that if you were looking at everything together, you would not be able to pick out these individual changes that might actually have huge implications in health and disease. It's kind of like if you're at a stadium and everyone's shouting, you're not going to be able to pick out what an individual person is saying, but what an individual person is saying might be very important. So this technology allows you to look at these individual cells one at a time and understand how they're working together and how they're, changing on their own.
Yeah, and I think Chris is I think there's a couple of really great questions and one on tools, but I just wanted to state the obvious, which is it is impossible to kind of overstate the importance of kind of the technological advances that really just accelerated our field, from the molecular level analysis to kind of network analysis of social networks and the computational approaches that are used there, the lessons we learned from mathematicians, and then the engineering developments. I mean, just huge, just breakthrough, really critically important. Chris? So.
Yeah, this conversation is awesome. You're all hitting on, I from my own perspective, but I assume everyone else's too. I mean, one thing that was raised was two things that I would just wanted to, nudge on. I tend to think one of the questions in the Q&A is about what would a high school student do as their extracurricular activities. I think motivation, fundamental motivation about the questions, finding them intrinsically, inescapably keep you awake at night, or at least before when I was young, that motivation, because that's the motivation, Marc, that then makes you go get the skill. And so in a way, if you had to put them in a hierarchy, of course you need both, but without one, you might not endure all the things you need to do to get the skill. And your acquisition of the skill might be kind of tone deaf. In other words, this is what an engineer is, not, I have to know how the brain works. So I might take these classes that don't make sense together in high school or in undergrad or in grad school or the books we all read. and then that's the thing that kind of, always having that as your touchstone feels really important in this.
Yeah, and actually, I want to point out that motivation, I don't think it has to be some super profound, realization in middle school that, you want to be a neuroengineer when you grow up or something like that. For me, that motivation, like I mentioned kind of in the introduction, was science fiction and watching, Star Wars as a kid and thinking that was really cool, it can just be cool. And that kind of gives you this drive to figure out, well, how could you make something like that be real? And engineering in a lot of ways to me is that learning the skills you need to be able to make something that isn't real. And that's really, I think, what drives me personally. And I think that, from that, middle school me who just was watching Star Wars or something, I don't think that I ever really planned out perfectly. I need to learn this, and then I need to learn that, and then I can be a neuroengineer. It was more just, you have this kind of idea of, oh, this would be such a cool thing to be able to do with my life. I don't even know what that looks like, kind of thing, but you kind of pick up those skills along the way. So in terms of explicit extracurricular activities, I mean, following your interest, I think, will ultimately lead you towards that goal. That being said, there are probably more specific extracurriculars you could do if you're particularly interested in this field. For instance, when I was in college, I spent a lot of time learning about the world of hobbyist electronics and Arduino microcontrollers and tinkering around with those. And I like to say that the best $45 my dad ever spent on my education was buying me an Arduino in college. And he might be pained to hear that after all the long, lots of money he spent before that. But it really was. I found myself kind of reading blogs and reading forums and tutorials and everything before my classes started in college. Just whenever I could get a chance, because I wanted to learn how to make a glove that I could use to control a helicopter or something. And I would say that those skills that I learned just from that extracurricular activity probably were the most valuable ones during my PhD that I used. And so, again, that was just by following my own interests, but it led me to where I was. And, certainly there are other paths, but I think that's kind of the one I took.
That's awesome. And I certainly agree that for any motivated student at any level, playing with Arduinos and just the wonderful explosive world of like even face recognition software, we've done that in some of our, it's really amazing. All right, I got a question for all of you now. That is from amongst many really great Q&A questions we're getting. So we're going to make this last 4 extra hours, and we'll release it on tape or something. But one question that comes up that seems to dovetail really nicely in this conversation is, so as tools become more advanced, allowing us to monitor activity at bigger and bigger levels, including the activity of large social networks, right? And the activity of, large sets of organelles and genetic networks, right? Do you believe that biological and cognitive sides of the question will become more closely linked, or are they inextricably oil and water, you think, or oil and vinegar, or in other words, are they such fundamentally different things that have to be pursued differently, or are they going to keep getting closer? Kaitlyn, please.
Yeah. So I'm going to say this first of all, as someone who works on pretty much the smallest amount of neuroscience, I'm working at the level of the individual cell. So as far as you can get from cognition, while still being in neuroscience, I think. But I think, as a neuroscientist, the fundamental belief is that the cognitive arises from the biological processes, and it isn't some completely separate thing that's happening. It's coming from the circuits. And I think Marc touched on this a little, but this is something I've touched on as well, is the amount of data we can collect is probably going to be what really breaks us through this biological cognitive divide, in that we are able to, at an unprecedented level, look at and recreate kind of all these different levels of the brain, whether we're looking at the transcriptome, whether we're looking at how the cells make connections, whether we're looking at the activity, and kind of being able to look at all of that together, I think is what's going to be able to push us through, that divide.
Other thoughts, Jay?
Yeah, so I want to jump in as somebody who's on the opposite end of this scale. So social networks are about as big of a cognitive question as you can get. And yeah, a lot of exciting techniques have been developed in the last, let's say 10 years, that allow you to use functional MRIs to look at people's brain activity as they're doing tasks inside of a giant super cool magnet, right? And so this gives you a sense for what the brain is doing as people are thinking. And that's really exciting. And the technology and the analytical techniques that we've developed have become so good that you can look at these tiny 3x3 millimeter cubes inside the human brain, and you can look at patterns in a brain region of these little cubes of brain tissue. And from that, by looking not only at the activity of the brain, but how it expresses that activity in patterns of activation, you can start to answer really interesting and fine-grained questions about, well, okay, it tells us something that this brain region activates, seems to be involved in thinking about people in this way, but we can do more now. We can actually go into those regions and say, in fact, when you are doing this specific kind of mental operation, we can see that reflected in this pattern of activity in the brain region. And so for us, this is actually one of our big goals, is being able to connect cognition and biology using these sort of new techniques. And so for us, it doesn't seem like oil and water. It seems just like, the question at hand, our next big project.
Actually kind of gets me thinking about a question I kind of think about a lot, which is the question of, is the brain deterministic? And so, this is a big one, but what do I mean by that? So, we like to think as engineers that we have this black box thing, and you put something in on one side, and as long as you put the same exact thing on one side, it always comes out the same on the other side. And the brain inherently, as we study it more and more, doesn't seem to be that way, at least not with the ability to look into what's going on that we currently have, with the tools that we currently have. It's interesting what Jay mentioned about fMRI. It's true that we have this really cool ability to ask someone to think about something, and we can identify the way they're thinking about it or what they're thinking about just by the way that some images light up on a screen for us. But if we were to put that same individual not in an MRI scanner, but instead, at their home or playing a sport, and have them think about the same thing, would those patterns look the same? And my thought would probably be no, and I don't know to what degree, but it gets this really interesting question of what level of fidelity do we need when we record from the brain to truly understand what's going on? And the brain is like, if you go deep enough, even past Kaitlyn's level of thinking of the single cell, ultimately the brain is this hugely ordered, complex thing that arises from something inherently stochastic, which is chemistry, chemical reactions that are by their nature random. It's the probability that two molecules will bump into each other, but somehow that creates this really complex behavior that at least has this appearance of order on top of it. And that's a really, interesting question to me. I don't know the answer to that of what we need to be able to truly be able to see and record everything from the brain. But
yeah, these are fabulous answers. These are really good. I mean, one thing I really like is, the question of the resolution of what we should record. Nested in that is the fact that the method Jay mentioned, which has been a revolution in brain science. I mean, it could not have been a bigger revolution than to look inside the human brain. And it measures maybe the most important network in the brain, which is the vascular network. So we don't even know the cell types. Although great work at RNA-seq and great work in all kinds of recording and doing that, but there's so many great frontiers here. This isn't Morley trying to find the last decimal point. We have a lot of revolutionary physics in that analogy to go through here, which is really nicely captured by your answers. So swinging it over to Diane.
Yeah, I mean, maybe I'll just add one quick thing. I know I'm a brain scientist and I'm at the Institute. I just fundamentally don't think that in the end, it is the biology that drives cognition. And I don't see the brain as being very different from the heart, as someone who started their work looking at cardiac function. And most of us who are kind of biophysics trained started in the heart and then moved into the brain when we felt we had enough control over what we were doing there. But like, you look at the EKG, it's a beautiful thing, right? But you would never in a million, like if you just looked at that, you wouldn't pull out all of the individual components, like the elementary channels that are, you know, working in coordination. I mean, it's just wild and crazy. We don't have that for the, I mean, okay, we have the EEG, but like not to at the resolution that you can get with the EKG. But like, just think what you can extract from the EKG now, like that it tells you which individual ion channel is like dysfunctional based on a change in the pattern of things. So I think that we're going there. It's going to take longer. There are layers of complexity here and more cell types and network. But like I fundamentally believe that that there's got to be that connection. But love the discussion. Fantastic. So I'm going to close with, I know it's shocking, but it's 4 minutes to the hour. But what I just wanted to is like kind of have you guys like leap into the future perhaps a little bit, or think about like, how do we, like you've really just been amazing. And I mean, I'm, you know, living what you were working on already. Like you just described things in such brilliant way. There's excitement and passion, of course, in whatever we're doing. We chose you carefully. no, like I just think that that's research and that's science. But what, you know, how do we think? How best can we think about the future? So the, you know, the great technologies that we have right now. Perhaps you could just say a little bit about how you think they are going to lead to this kind of introduction of new treatments, technologies. We all don't have to be translational. Fundamentally believe in basic research, but like if you leap forward 20 years and think about what you're doing now, can you give us a little insight into what the future is going to hold? And you literally have like a minute each, 60 seconds. Who wants to start?
I can go first.
Go, Kaitlyn.
In a minute. But I think kind of the things I'm working on with reprogramming and basically being able to generate large amounts of human cells in a dish are going to absolutely change how we even go about drug discovery and treatment, because human aging is obviously very different from rodent aging, and we're finally going to have the material we need to test these things in a way that we have not been able to test on humans, because that's obviously unethical in the past. So I think we're due for an explosion just because of the ease of these experiments and how they're just going to get easier in the future.
I guess I can go next. So I really do exist on this edge of translation. So I think a lot about where we will be in the not too distant future with that. And aside from the big elephant in the room of the FDA, which, you know, 20 years from now, even stuff we're working on now might not be in people, but I think the way that we're pushing towards, translatable technologies, and as we get these abilities, that I've kind of been alluding to this whole time about being able to access even more of the nervous system, we can start to hopefully answer these questions of, you know, how does the brain encode certain things, and go from this era of, you know, DBS for Parkinson's, where we don't truly understand how it's working, but it kind of does, to truly understanding how it's working, and then being able to push forward. And once we get to that point of that understanding, that's when we can really develop targeted technologies to push translation.
Yeah, I really like that idea. I mean, for us, you know, like this is, the translational benefits are probably far, far into the future. I can't even anticipate what they're going to be. But as basic scientists, you know, what we're trying to understand is, what about people's brain activity, about the coupling of brain activity to behavior leads people to be lonely, or to be ostracized, or to be popular, or to be a social butterfly. These, I think, are questions that will help us just better understand our human experience. And, you know, I think there's some intrinsic benefit to understanding these questions. And who knows where this technology will take us, right? Or who knows what kinds of explanations that we'll have about, you know, just being to tell our own story about why we are the way we are. So for me, that's what the future of brain science looks like. Fabulous.
Thank you. we hit the hour. Incredible as that is. So I really, Chris and I both thank you all. That was fun, interesting. And as I said at the beginning, I think you proved me right, which is, if you want to know about, the exciting things that are happening, listen to eloquent explanations of complex research topics, go to the early stage scientists, because they are where the future, I mean, you are our future, and I'm really excited by that. Listening to all three of you, I think, you covered an incredible breadth of topics. And I have to say, what surprised me most was the way that you were all thinking about how do you create an environment to really enable great research and innovation and creativity, which is what we talk about all the time. Honestly, I've learned a lot from listening to you guys. And I also, was thinking, Jay, as you were describing this, I mean, maybe we can come up with a formula an idea of like, who is the right person that we should be recruiting next. I just love that the reductionist approach, but of these complex issues, but like deeply thinking about the community and the environment that we're in. So thank you for that. It was tremendous. Chris, do you want to say anything to close? And then we're gonna thank everyone. And
yeah, we have to quote the final Q&A entry is not a question, but brilliant panelists.
There you go.
Enlightening listening to all of you. So
There
make
you
sure
go.
to feedback from the listeners.
So I hope you all, thanks for joining us. I hope you'll join us again for the next Carney Conversations. I can tell you one of the things on the schedule is thinking about, how we function in the uncertain world that we're in right now. And so we've got a couple of very exciting panelists lined up for that, but there'll be more information coming in your email soon. Thank you everyone for joining us. This was tremendous. Thanks, Kaitlyn, Jay, and Marc. Take care.